Overview today, we're going to do some intros, a little bit of logistics and overview of the course. And again, there's more detailed information on the video and in the syllabus. We're going to talk a short history of epidemiology, a little bit about infectious disease EPI in 2020, which is obviously a huge topic right now. And I'm going to talk a little bit about the readings. So I have put a short assignment on Moodle asking you to introduce yourself. Please, everyone, fill it in as soon as possible. It's just so I can tailor the course to meet your needs. This is still sort of a work in progress since I have never taught this course at UMass Amherst, and so I'm creating a new course basically from scratch. So again, this is also in the intro video, but there are two sections of this course, and it's somewhat confusing. I hope it doesn't become very confusing over the term, but one is an online course and the other is a residential course. The online course is fully online. There are no Zoom meetings, no synchronized learning. You get weekly assignments and readings, and you do the work weekly, and then the major assignments you do on your own as well. So, but the residential course was originally intended to be met in person, but because of COVID-19 and all the things related, it got pushed to an online format. So the residential course is the same content as the online course, but has the added aspect of having Zoom meetings that are scheduled and students need to be present. So check the section you've signed up for and confirm if it's the online or the residential. And if you have any questions, please email me. The workload for both courses will be essentially the same. There's just things that are done online pre-recorded for the online version and live over Zoom for the residential version. So please feel free to ask me any sort of questions about this. I hope this is not confusing. So all course information will be on Moodle. The syllabus will be revised regularly. Going to sort of play it by ear. We have a schedule of topics that we're going to go through, but there might be some sort of minor changes that happen along the way, just based on student reaction and student concerns or needs or whatever, or my concerns and needs. So it's just really I'll try to keep people up to date on what's differed, whether something gets changed in the syllabus. And I'll warn you ahead of time, it's not going to be any major changes, no new giant assignments. There'll be nothing, no big, huge, disastrous surprises is what I'm saying. Readings, I'm taking a little liberal view of what a reading is. And they mostly consist of selections from the textbooks. We're using the Giseki text and reading a book called The Wisdom of Horrors in this course, and there'll be selections from that, basically one chapter a week, roughly. I'm going to be posting publications from the scholarly and popular literature, and also I'll be posting video and audio content, which technically aren't readings, but you still will have to read and watch them, and I hope they're interesting for you. I think that, especially when you talk about infectious disease, seeing things really helps to drive home, certain aspects of infectious disease, particularly when we're talking about global health and areas of the world that people may not have been. It helps to see what is going on in these places, because that gives us some answers on why transmission may be occurring, because infectious disease is, we are part, of the cycle of host and pathogen. We are the host, and how we behave directly impacts how the pathogen can affect us. And so, things like hand washing, for example, places, where people regularly wash their hands and have access to clean water. We have very different dynamics of transmission for things like in waterborne diseases than areas which do not have access to clean water, for example. Vaccines are one. This is another thing that we've created, and some people have access to it. Some people have opposition to it and don't want it. Some people have no access to it, and this changes the dynamics of transmission. So we're going to be talking a lot about that next course. I am sort of, I'm an epidemiologist, but I consider myself to be somewhat of a sociologist in some sense. I'm definitely interested in social determinants of health and behavioral determinants of health and infectious disease transmission. So we're going to see a lot of video and audio throughout the course. So with the lectures, in combination with the lectures, the readings will provide a basis for all quizzes and tests. When we have, quizzes, I mean, there's just things that come out of the lecture, and it's basically a way for me to get you to go back through the lecture and really pay attention to what, what information is being provided to you. They'll be put on Moodle, and they'll just be, something you fill in on your own on Moodle by the due date, of course. Regarding the readings, specifically the publications from the scholarly and popular literature, you will write a weekly mini reflection on one of the readings, which will be specified on the Moodle site. So basically, I'll tell you, write your mini reflection on, this paper that I've posted. And just basically, you tell me what it's about. I'm going to write a little document on Moodle. It's going to tell you how to do this. It's just going to be, what's the paper about, who is the study population, what sort of study designs they use, what is the pathogen of interest, and what are the methods and conclusions, these kinds of things. It'd just be simple, 250 word, mini reflection, really easy. And we will do that weekly, and I'll be posting something on the Moodle site about this. Major assignments of the course will consist of a primary literature presentation, and I talk about this on the video. So the primary literature presentation is going to be for all students. The fully online students will pre-record their presentation. They will talk to a camera because I want to get you used to communicating about these things. And I said camera, but you can do this audio if you want. I prefer that you do it on camera, but if you don't want to, that's fine. You can just audio, do it over audio, and that's fine with me too. Because I want to get you in the habit of being able to communicate these things to other people. Because it's part of what you'll be doing as public health professionals, is communicating complex concepts in ways that people can understand. That's part of our job as public health professionals and epidemiologists. What we do directly impacts people's lives and well-being, and they want to hear from people like us what we think and what we know, and they want to hear it in a way that they can understand. So there's just going to be one of these. You will be assigned or pick a paper, and you'll do a 10-minute video or audio recording for it. For the residential students, they'll do this over Zoom live for the other students to listen to as well and take questions. So more on that later. There's going to be a midterm report. It'll be a 6-page paper on infectious disease of your choice. We're going to talk about this a little bit next week to give you a heads up on it and start signing up for, things that you might be interested in or taking suggestions. You will pick a pathogen, and you will write a 6-page report on it. It should be very interesting for you, I hope. Double-spaced, of course. And then the final exam, take home in this kind of context is sort of, really a sort of catch-all to mean that you're going to use any of the materials given in the course, to do the exam. So you're going to be applying newfound knowledge to questions from the primary literature and over the things that we've talked about over the course. That's a long way away. We'll talk about that. Please write me if you have any questions at all about the material or the requirements for the course. My email again is plarson@umass.edu or anfangen@umass.edu. Either one, you might get a reply from me from either address. So now, what I'm going to do is take you through a basic history of epidemiology. What is epidemiology? The word epidemiology comes from the Greek words epi, meaning on or upon, demos, meaning people, and logos, meaning study of. So we're studying the things upon people. The epidemiology, the sort of accepted definition, is the study of the distribution and the determinants of health-related states or events in specified populations, and the application of this study to the control of health problems. And this is from the CDC. So basically, you know, we talk about who gets sick and why, and how bad is it over the population. So who gets sick, why they get sick, and what is the extent is really what we're interested in. Epidemiology is a broad field. It basically encompasses everything. We have a little bit of medicine, a lot of medicine. You know, we have a little bit of sociology, we have some anthropology, we got statistics. We basically have everything in there. So if you're interested in a wide range of things, it's really really a wonderful field to be in. That's why I chose it, because you know I like to look at a lot of things at once, to keep myself sort of excited about all different kinds of things and look at how all these things connect together. So, yeah. It's fairly new. Epidemiology is not old, you know, in comparison to some things like medicine, for example. But it has a long and interesting history. And so right now I'm going to talk about some major events in the history of epidemiology and public health. So the first stages of epidemiological thinking, you know, first John Grant, he wrote this index on the bills of mortality, natural and political observations, and basically, you know, we did the first sort of like you know survey of why people died, and it's quite fascinating looking at something, you know, from the 17th century, looking at numbers of people who died in cities and neighborhoods and why they died. It's quite fascinating to go back that far. So he was looking at data from London mortality reporting from 1604 to 1660. He was especially interested in plague, the Black Death, and developed the first life tables, first demographic life tables. And he found plague underreported by 25% and examined relative mortality in plague years and discussed things like epidemic and endemic diseases, and also suggested underreporting of syphilis and increases in rickets. So it's quite fascinating. You know, when he started writing these things down, he found that people were reporting much less than there actually was. So this is quite fascinating. So smallpox is a horrible disease that we're going to talk about a little bit later. It's unique among all of infectious diseases, in that it's not with us anymore. And it was a disease where, you know, a pox came across a person's body and had about a 35% mortality rate. Really just terrible. And if you read the descriptions of the disease itself, it's just absolutely horrific. So we can be happy that it is not with us anymore, because I can't imagine that we would have the kind of society that we have now, you know, if we still had smallpox around. It's quite bad. At least, you know, not to the extent of what was you know, back in the 1800s and 1700s. So Edward Jenner noticed that dairy workers rarely had smallpox after having pustules from a related virus, cowpox. And so he would, you know, take some pus or, you know, a scab from cowpox and inject it, you know, rudimentally into a human. And he tested this out on his neighbor's son, which would be impossible today. And, you know, fortunately the son didn't get smallpox. Before that, you know, he would practice this thing called variolation, which is quite ancient actually, where they take a little bit of pus and you know, inject it into a human, And so that would sometimes create disease, but sometimes create some sort of resistance within the body, so the disease wasn't as severe when they did get sick. So, but generous activity was really sort of the first formal vaccine, if we can call it that. And his first sort of formal like clinical trial on his poor neighbor's son, James Phipps, age 8, who arguably was one of the most important people in the history of epidemiology. So William Farr used smallpox data, compiled law of sickness, which are rates per 100,000 people at every stage of infection, sick, recovered, or dying at 5-day intervals, and worked at the registrar's office collecting national morbidity and mortality statistics. So, I mean, he was, sort of one of the first people to break down things by population like that, standardized numbers. John Snow is very famous. He proposed water contamination as a source of an 1854 London cholera epidemic. Cholera is a waterborne disease that causes terrible bouts of diarrhea, and basically you expel most of the fluids from your body and die quite rapidly. It's a really quite horrible disease. But Snow was famous because he started plotting out the locations of where people were getting sick, and figured out that they were all using the same water pump, and famously saved the day by going to the Broad Street pump and turning it off. And supposedly that stopped the epidemic in London. Whether this is true or not is up to debate. Whether it's true or not, it actually stopped the epidemic is up to debate, but it is true that that pump was a source of cholera infection for a lot of people, for sure. So not only is this one of the sort of first epidemiologic studies, it's also one of the first sort of GIS study, Geographic Information Sciences study, where they used a map to isolate the point of infection, the location of disease. Francis Galton, another, old white guy like this, created statistical methods like regression to the mean, correlation, and pioneered the development of questionnaires and surveys. Carl Pearson, another older guy here, also, came up with the birth of modern statistics. Statistics is also quite young. And both of these guys, had a great impact on medicine and epidemiology. And the idea, importantly, that health can be quantified. We can use data to learn something about health. That was really what their great contributions were. Robert Koch came up with what's called the Koch's postulates. He was one of the fathers of bacteriology back in the 1890s. Before this, it was sort of, not well understood why disease occurred. There were sort of a number of ideas. One was the miasma theory, where there's bad air from swamps that is making people sick. There was ideas of the humors, all different kinds of ideas that, some part of the body was out of balance and needed to be put back into balance, and then, disease would be cured. But Koch, was one of the first guys to start isolating, starting what's called the germ theory of disease, where diseases are caused by microorganisms that enter the body and use the body for replication, and also cause disease in that process. So he came up with what's called Koch's postulates. The microorganism must be found in abundance in all organisms suffering from the disease, but should not be found in healthy organisms. This was really important. The thinking is that if we found the pathogen in sick people and we didn't find the pathogen in healthy people, then the pathogen must be causing disease. Of course, now, we know more than this. We know that there are things like asymptomatic carriers of the disease, who have the pathogen but do not have symptoms. We know that now, but back then, this was sort of revolutionary. The microorganism must be isolated from a diseased organism and grown in pure culture. And this is, for bacteria, this is quite important. I mean, we take a blood sample, we isolate the pathogen, and if we're able to replicate it and grow it in a petri dish in a lab, then this is one of the requirements that we needed to satisfy that this pathogen was indeed causing disease. The cultured microorganism should cause disease when introduced into a healthy organism. And this is, part of where we come up with animal trials. So for example, like rat models, we take the pathogen from a human, grow it in a dish, put it into a rat. And if disease was caused in the rat, then, we might start assuming that this is a pathogen that causes disease in humans as well. Of course, we know now that not all pathogens cause disease in all types of hosts. There are some pathogens that only cause disease in humans, some pathogens that only cause disease in animals, and we know that now. But back then, this was a revolutionary idea. The microorganism must be re-isolated from the inoculated diseased experimental host and identified as being identical to the original specific causative agent. So that's a sort of, the back-end confirmation. Did the pathogen replicate within inside the animal? If yes, then all these four criteria are satisfied, and we can assume that this pathogen is in fact causing disease. Ronald Ross, he won the Nobel Prize for first identifying malaria parasites in mosquitoes fed on patients. Before that, people did not know what really caused malaria. They associated it with swamp gases or bad air, referring back to the miasma idea that I was talking about a minute ago. People did not really understand that mosquitoes were transmitting malaria. So he was the first person to open up a mosquito and find parasites that were picked up from humans. And parasites they were going to deposit within the humans. We'll talk about malaria later. Malaria is incredibly complex and quite fascinating. So we'll definitely go over this later. He is also important because he developed one of the first transmission models, which is an idea that we're going to talk about later, about representing infectious disease transmission using mathematical models. He did this in 1911. It's a fascinating paper that he wrote on this. His models are quite fascinating and incredibly complex. We'll look at it later for sure. And this allowed relative contributions of treatment versus vector control to be gauged. So, how much treatment do we need and how much sort of, vector control, mosquito control do we need, or do we need a combination of the two? So we're going to definitely talk a lot about malaria later because malaria is one of my favorite diseases. It's, epidemiologists, we have favorite diseases, which, of course strikes lay people as being quite strange, but I hope, over the course of this semester, you find some of these diseases, these pathogens as fascinating as I do. Ronald Fisher had the birth of statistics, single-handedly developed much of modern statistics and quantitative biological science. Most of the tools that underpinned all quantitative analysis were developed by him: randomization, design of experiments, and maximum likelihood. We use tools developed by Ronald Fisher every single day right now in epidemiology. Really incredible contributions there. Yellow fever was a disease also transmitted by mosquitoes, and William C. Gorgas was one of the people that identified Aedes aegypti as the primary vector of yellow fever and led to programs to eradicate yellow fever, and also led to the development of a vaccine. Because during World War II, It was important to have, if you're going to send soldiers to, hot areas of the world, it was important for them not to get sick. So you'll hear over the course of the term that, the military has driven a lot of vaccine development for what we call the tropical diseases. You'll see that a lot. It's not, not on the side of the military or anything like that, or on the side of warfare or anything like that, but historically that's been the case. Polio. Polio is another waterborne disease that at one time caused paralysis in people. Like my grandfather had a bad leg from a bout of polio when he was a child. And you can see that, we look at the graph here, we can see the cases, paralytic polio cases were very high, around the 30s and 40s and 50s. And then drop off to nothing because we have a vaccine. There were two vaccines. The major one was the Salk vaccine, which was 80 to 90% effective in preventing paralytic poliomyelitis, and remains the largest public health study ever. It's really quite fascinating. Now, most of you have been vaccinated for polio, and polio is really foreign to us, in the United States. For people from developing countries, it's still not uncommon to see people who, suffer from lifelong paralysis from bouts of polio that they had when they were children. But cases of polio are definitely declining across the world, and one day there might be no transmission of polio at all. We'll talk about eradication versus elimination later in vaccines as we go through the course. George MacDonald, more malaria work, expanded Ross's models, formed the basis for planning the global malaria eradication campaign. We're going to talk about eradication later, but for a number of the diseases historically, there's been a push to stop transmission overall, to stop it forever. And there was a long push during the 40s and 50s to eradicate malaria, stop transmission completely. But MacDonald's contribution was to create equations like this to describe things like R naught. You'll see R naught referred to a lot in COVID-19 world as the number of cases that arise from a single case of the disease. And so malaria is quite complicated. It takes many parameters, how many times a mosquito bites, how large the human population is, how many mosquitoes there are, all these different things put together, form the R naught. And we're going to talk a little bit about R naught later and why it's a much more complicated number than how it's presented. But yeah, R naught, number of cases arising from each single case of infectious disease. Global malaria eradication was greater understanding of disease dynamics suggested malaria eradication was feasible and vectors were the key. This global program started in 1955. There was elimination in many settings, so local, cessation of transmission, but insecticide resistance stopped the program in 1970. So basically, many of you may have heard of what's called DDT. And DDT was used initially to control mosquito populations, but it was quickly found that mosquitoes became resistant. That is, DDT stopped working to kill the mosquitoes, and they had to stop the program because the overuse of DDT was also leading to large impacts among bird populations. Smallpox. We're going to talk more about smallpox later, but after the development of the smallpox vaccine, we successfully eradicated smallpox around the world. There's no transmission of smallpox anywhere in the world now, not since what was this last year? I think 1980 something was the last case. Doesn't say here. That's okay. But last case of smallpox in the United States in 1952, considered to be eradicated. And Africa was much later. It's harder to deliver vaccines in resource poor areas than it is in places like North America. We are fortunate that smallpox is no longer with us. Smallpox is considered to be eradicated. That is, transmission does not occur between humans anymore. But samples do exist in labs in Russia and the United States. So the pathogen is not completely gone from the world. It's there, but there's just no transmission right now. So this brings us to a question. Could we have beaten infectious disease? So in the 1970s, 60s and 70s, we had things like antibiotics. We had large numbers of vaccines. And the thinking at the time was that infectious disease would no longer be a problem. We would have the technological tools to deal with any type of infectious disease. And so future generations of people would not have to worry about dying or getting ill from infectious disease at all. And Dr. William Stewart, the Surgeon General of the United States from 1965 to 1969, is famously said to have said, "It is time to close the book on infectious disease." Now, it turns out he didn't actually say this, but this was the thinking at the time, that we're done and it's time to move on. So like I said, 60s and 80s was very, very quiet. I would really sort of say that 60s, to the early 90s was very quiet. It really wasn't until we started, seeing things like HIV. When HIV appeared in 1981, it was a huge wake-up call for public health in general. Colleges like Yale University, for example, were closing down their infectious disease departments because they figured that we were done. Then HIV came around and raised all the, the warning signals that no, we're not done. There are new infectious diseases out there that we don't have a cure for and that we can't find a cure for. We still, to this day, do not have a vaccine for HIV. Because of its unique way of hijacking the immune system. And we probably will never have a vaccine for it. We have medications to keep people who are infected, from developing serious disease, but we don't have a vaccine. So still with us, it also has to be said that, dengue fever is also another pathogen that's reemerged or back in a major problem in places like South America and Central America. Ebola was one that maybe you are all familiar with that came out and especially in 2014 killed more than 14,000 people in West Africa. And so especially now today where we're basically watching a disease pandemic in motion, COVID-19 is another one. Infectious diseases did not ever go away. We just got a little bit lucky in that small window between the 60s and the 80s. When you read the Ringo paper, you'll see sort of some argument that we need to start thinking about infectious disease again. Because there's all this effort being put into chronic disease, thinking that infectious disease is going to go away, and not enough attention on infectious disease. So Ringo was making some predictions about infectious disease epidemiology, but there's always been, at least since I've been in the field, this sort of like conflict between chronic diseases and infectious diseases, as to say those two things are mutually exclusive. But we found out now that they are not. That some pathogens actually cause cancers, like the human papillomavirus, for example, causes cervical cancer and oral cancers, which we know that. Helicobacter pylori causes ulcers in the stomach. So chronic disease and infectious disease really are not two mutually exclusive things. You know, when you go out into the world, you know, you might start working with chronic diseases because many of you might be working at local public health departments, might be working with things like obesity, cancer, these kinds of things, but there are actually some infectious determinants of this, and it's becoming more and more apparent that these two fields really are not all that separate. HIV pandemic, more and more money has been spent on HIV and AIDS because we have put tons and tons of resources into it to dealing with HIV, AIDS, coming up with treatment regimens, antiretrovirals, drugs, drug distribution, trying to make them available to areas that need them, for sure. We're going to talk a little bit about funding for infectious disease as we go on in the course, because this is also a major issue. You know, what diseases get funded? What gets paid attention to? Does the funding actually have an impact on the disease itself? So we're going to talk more about that later. So late 20th, early 21st century developments, Lyme disease is one that I think everyone is familiar with, especially out for y'all out in Massachusetts, New England. Lyme disease is a huge problem out there. SARS, and of course, SARS-CoV-2 that we're dealing with right now, the Nipah virus, Ebola, West Nile, MERS coronavirus, and of course, we have to put COVID-19, SARS-CoV-2 in this list. Recent developments, you know, Zika is one that we're seeing right now. We have problems with drug resistance, vaccine hesitancy, and we're going to definitely talk about this. Emerging infectious diseases, EIDs, stalled progress on old diseases that we're going to talk about when we talk about the big three: TB, HIV, malaria, polio, and guinea worm disease. A lot is going on in infectious disease right now. Very, very recent, SARS-CoV-2 appeared in January of 2020 in Wuhan, China, but may have emerged previously. We don't actually know when it actually emerged. It's highly infectious and causes severe disease and mortality in some persons. COVID-19 has had wide-ranging impacts on health, public health, medicine, economics, and culture, as we all are seeing right now, every single one of us. We're going to talk about COVID-19 later in the term because obviously it's topical and of interest to us. We're going to talk about why it emerged and situation in Wuhan when it first emerged, and then how it came to the United States, and how it's transmitted throughout the United States, and what impacts it has. A lot of which you are seeing in person, because we're all taking a course online. Some of us were supposed to be in a classroom, and we're not because of COVID-19. Okay. So communicable versus infectious. So there's sort of two different terms. Infectious is caused by organisms. Communicable is considered to be a subset of infectious by Giseki. Diseases that can be transmitted person to person by any means. And transmissible means person to person by unnatural routes. These are just sort of definitions. Weber calls all diseases communicable, infectious, microorganisms transmitted person to person. And CDC listed infectious diseases include malaria, dengue, and Lyme disease, which are all vector-borne disease. So as we're going to talk about later, not all infectious disease is transmitted directly from human to human. There can be another organism in between, or there can be an object in between. We're going to talk about different transmission pathways later. And skip this. Core semester reading. It still says The Origin of AIDS. I'll have to fix that. But we're actually going to be reading The Wisdom of Horrors, which is available via the UMass Library. It's a great book. This book just talks about the wide-ranging impacts of HIV and AIDS and policy and business. And it's really a fascinating, wonderfully written book. So I hope you really like this. I'm going to read a chapter a week. I'm going to have a little quiz on it just to make sure you're still reading. But yeah, this is going to be a hopefully a good read for all of you, for sure. Students from other departments or schools, I hope that all of you have had an epidemiology course at some point. Some of you might have used Gordis Epidemiology. This could be a good background for the course to use while we're going through it. It's not required at all. This is usually just a text that people use in other schools. Some people might have used Kenneth Rothman's text, Intro to Epidemiology or Modern Epidemiology, if you're taking an advanced course. Those are all good. So yeah, you know, I mean, we're going to be like sort of coming up from the ground up here, not going to be talking too complicated from the beginning. But you know, having a background in epi is gonna be good for you. So if you have not had an epidemiology course, please contact me and we'll see what we can work out. Okay, so that is it for today. I'm going to post this to Moodle, which is the only way you will have seen this. So if you have any questions, please email me. Yes, thank you. And please enjoy the readings.